Hello everyone. Today we will be discussing about the characteristics of the PBR322, which is commonly used in the biotechnology. Now, what is the size of this is nothing but 4.3 kilobase. Two sets of the antibiotic resistance it consists. Therefore, selection is a two-step process. Reasonably high copy numbers of the are produced. Selection using antibiotic resistance. Ligation of the alien DNA is carried out at a restriction site present in many of the two antibiotic resistant sites. See, everybody knows that PBR322 is the one which is having a structure with the ampicillin resistance and also tetracycline resistance site. So, how can you see this? See, here we see the different types of the structures parts of this, right? So, here E. coli 1, that is Escherichia coli 1, and these are the proteins Caryophyllin latum and also Haemophilus influenza 3 and also Bacillus ameloefficacens and also Streptomyces album and also Proteus vulgaris and Providentius tuberti and also Proteus vulgaris. These are the proteins which are present on that, and also ampicillin resistance sites and also tetracycline resistance site, and it is having origin of replication also. And what is this? ROP stands for, it is a protein of replication also, we can say. And because of this reason we can use in antibiotics. So what we are going to do is ligation of the alien DNA is carried out at a restriction site present in one of the two antibiotic resistant genes. So you have seen that ampicillin as well as tetracycline. So in any one of this region, you are going to cut. So either ampicillin or either in the tetracycline. So you are going to introduce a new foreign DNA. So you can ligate a foreign DNA at the BIMH1 site of tetracycline resistance gene. So in the vector PBR322. See, now here you have seen the PBR322 with the strands. Like here you can ligate the two and you can remove the tetracycline part also. Then the recombinant plasmids will lose tetracycline resistance due to the instead of foreign DNA, but still it can be selected out from the non-recombinant ones by plating the transformant and ampicillin containing medium also. See, when you're placing this kind of the things, PBR3, the transformant growing and the ampicillin containing medium are then transferred. So, medium containing tetracycline. Then what happens here? Here, the recombinants gene and medium ampicillin medium but grow on the medium but not on the containing tetracycline, but non-recombinant will grow on the medium containing both the antibiotics. So in this case, one antibiotic resistant gene helps in selecting the transformants, whereas the other antibiotic resistant gene gets inactivated due to insertion of alien DNA and helps in selection of recombinant DNA. So this is about the advantage. And what is the disadvantage? Selection of recombinant due to inactivation of the antibiotics is a cumbersome because difficult procedure, because it requires simultaneous plating on the two bloods having different antibiotics. So replica plating method will be done. So this is about the PBR322. So in a simple way to talk about this, see students, here it is a PBR322. Now here it is having the tetracycline resistance gene, and here it is having the ampicillin resistance gene. Now what you are doing, you are introducing the foreign DNA into the tetracycline area. Now what happened? It get inactivated and it doesn't produce any resistance for the tetracycline. So because you are introduced the foreign DNA, it is a foreign DNA. Now when you place in the medium containing only ampicillin, okay, and in that the plasmids are able to grow in this medium. When you place in the tetracycline medium, this will not grow because they do not have the resistance power to tolerate the tetracycline medium. So this is what we need to check both the stands in the ampicillin medium and also tetracycline medium. So it is a cumbersome and it is a very difficult process. To solve this only, another technique, another plasmid can be isolated, that is called as a PUC8. PUC stands for, P stands for plasmid, U stands for university, C stands for California. So where it is extracted, 8 is the strain number. Now what is the interesting point to note it down is ampicillin resistance gene it is having, and at the same time here there is a lax Z gene also it is having, which is helping for synthesizing the beta galactosidase. and at the same time it is having the E. coli also, salmonella, so I write streptomyces also, and also bacillus liquefaciens, and also streptomyces albus, and also providencia tuberti, hemophilus influenza, and everything it is having. So what is advantage? High copy number, that is 500 to 700 copy number it will produce. Identification of the recombinant cells achieved by a single step also, that is plating cells into agar medium containing ampicillin and also exgal. Chromogenic substrate for beta galactoside enzyme encoded by the LACZ. And what is the main important steps to note it down here is, see, when you apply exgal, it is a chromogenic colorless chromogenic substrate, which is actually 5-bromo 4-chloro 3-indolyl beta galacto pyranoside also we can call it. And generally when you are applying exgol to this medium near the lax jet gene, then what happens? Lax jet is the one which is producing beta galactosidase. When beta galactosidase is producing, it is hydrolyzing the catalyzes the hydrolysis of the X galactosidase. Then what happens? It is turning to blue color. So this indicates that LAC-Z gene produces the beta galactosidase. Now what you are doing? You are completely removing this particular LAC-Z and you are introducing into the foreign DNA. Then what happens? LAC-Z gene lacks the get inactivated because insertion of the foreign DNA. Then what happens? What happens is the LAC-Z gene do not produce the beta galactosidase. In turn, X galactosidase will not turn the blue color. Do you understand what I am telling? See, when LAC-Z gene produces beta galactosidase, it turns the colorless X galactone into blue color. But after introduction of the foreign DNA, here you are introducing the foreign DNA. Then what happens? Here LAC-Z gene get inactivated because of insertion. Due to that, we call it as insertional inactivation. Who got inactivated? LAC-Z gene. Who got inserted? Foreign DNA. You understand? Now, LAC-Z gene do not produce a beta galactosidase. Because of this reason, we call it insertional inactivation also. And later on, and xgal, when it is present, it will be colorless only. It cannot produce into the blue color. So this is the very simplest, because only it is having ampicillin resistance side, and we can develop in the medium which is having only ampicillin. So this is one of the advantage of the PUC 8. Thank you.